Good afternoon, everyone, and welcome to the UCLA Ethics Lecture, which today is the Weinberger-Vermouth Family Genetics Ethics Program. And we share this with CEDARS. We're able to do this annually, and by my count, I think this is the 10th Genetics Ethics Lecture. Clayton, Botkin, Junst, Greenlee, Knopper, Cook, Deegan, Murray, Olshansky, last year Susan Wolf, and this year we're lucky enough to have Jonathan Kimmelman. This lecture happens only because Tom and Leslie are willing to bring in a speaker, in this case internationally, so that we here at UCLA and those at CEDARS can get together, learn about new work that's being done, have rowdy discussions, and talks get given both at CEDARS tomorrow and today here at UCLA. Dr. Kimmelman is a particularly interesting person to bring in for a talk like this because his work is the kind of stuff that we do quite often. That's drug development and trying to translate brand new molecular findings to the bedside. He's associate professor in biomedical ethics and social studies at McGill, but he also is in the experimental medicine, epidemiology, and biostatistics, and occupational health and human genetics. And getting your merit increases must be really interesting. He got his PhD in molecular biophysics and biochemistry at Yale and joined McGill in 2005. His research looks at ethical, social, and policy challenges in testing novel medical technologies in humans. And in fact, in the ethics committee last month, we reviewed a paper of his that looks at specifically how we develop new trials and whether we imbue them as ethically as we might. He chairs the Ethics Committee for the International Society of Stem Cell Research, and he's a member of the Gene and Stem Cell DMSB for NHLBI, which must be quite interesting. His book in 2009 from Cambridge Press is Gene Transfer and the Ethics of First-in-Human Trials, Lost in Translation. And today he's coming to us to talk about how many patients does it take to develop a new cancer drug. Dr. Kimmelman.
Well, first I want to thank the organizers for the invitation and to the funders for this wonderful opportunity, and I truly am honored and flattered to be in the company of some of the speakers you've had before. Second point I want to make really quickly is it's amazing how everyone's sitting here on the left. My wife is a pianist, and usually at her concerts people always sit on the left so they can see the keyboard, but here I can't really, there's no obvious explanation for that. Okay, so let's get started. So I'm going to start with a prologue. Probably most people here are familiar with some really fundamental tenets of research ethics. One of the sort of foundational elements whenever you do research in human beings is that risks always need to be balanced and exceeded by the prospect of benefit in clinical research. And typically the way that is cashed out when we do research in human beings is by ensuring that risks to human subjects are minimized and that those residual risks left over are acceptable against the benefits. Now, there are two, I think, serious problems with the way that we typically try to operationalize the requirement to minimize risks to subjects in human research. The first one is that typically when we think about minimizing risk to subjects, we think about it on a per-subject basis. So if you're doing a clinical trial and you can get the information you need with one lumbar puncture instead of two, you minimize risk by doing one lumbar puncture. Likewise, if you can do a partial burr hole in a sham surgical procedure to the brain, for example, you use that instead of actually injection to the brain. But there's another way to understand the commandment to minimize risk, and that's namely to minimize the number of subjects that participate in clinical trials. Now, this is certainly something that is, occasionally kind of surfaces with research ethics committees and the design of studies, but it really does not command the kind of influence I think it deserves when we think about risk-benefit in clinical research. So the second major limitation has to do with the unit against which we are assessing risk and benefit. Typically, when we're looking at an individual clinical trial, we might be looking at the prior evidence directly preceding that trial and making some judgment about that protocol, whether it is well justified against the prior evidence so that it can support the study that comes afterwards. But what I want to suggest to you in the next 30 to 40 minutes is that if we're actually going to be assessing risk and benefit of a given protocol, we have to evaluate not merely the trials that directly precede it, but some of the prospects of trials that might follow from it, as well as trials that are being pursued simultaneously, trials that are actually related to the individual protocol. The consequence of these two oversights that I'm going to describe to you in the next half hour are that we have a large amount of inefficiency in the way we develop drugs, a moral inefficiency, where I'm understanding moral inefficiency in terms of the amount of patient welfare that is ultimately sacrificed in order to generate usable information for healthcare systems. And we have an excessively large amount of residual information that is generated in clinical research that really doesn't have as much clinical utility as we would like. And the consequence of that, in the context of the fact that most drugs that are put into clinical development are not successfully translated, is that many more patients than necessary are exposed to risks and burdens in the process of developing new drugs, that our resources, in particular our human capital, are misallocated towards flagging research trajectories, that we spend a lot more money developing drugs than we need to, that money is ultimately absorbed into the cost of pharmaceuticals, so this ultimately burdens healthcare systems. And so what I'm going to be describing in this talk is trying to address the question of how much patient welfare is sacrificed to develop a new drug, where are the excesses the greatest, and what can we be doing to minimize those excesses. My talk is going to divide into 6 different chapters. First, I'm going to describe some of the methods that I've used to look at moral inefficiency in the context of research. I'm actually going to be presenting data to you today. Then I'm going to show you how I've applied those methods to study single drug development trajectories for cancer drugs. Then I'm going to describe how I've used those methods to look at groups of cancer drugs that are being developed, that are successfully translated. I also want to apply the methodology to unsuccessfully developed drugs and compare and contrast successful with unsuccessful drug development trajectories. And then I want to ask whether or not the findings that I've generated are generalizable outside of cancer to realms like neurology, for example. And I'm going to close by unpacking what I think are some preliminary moral implications and policy implications of the way we do drug development. Let me first start with the methods. As I said earlier, what I'm primarily interested in is the moral efficiency of drug development, where I understand the inputs, the most morally relevant inputs are the patients that we use to conduct research, and the most morally relevant outputs is the information that can ultimately guide healthcare decision making. And in order to empirically study this moral inefficiency or efficiency, what we need to do is to look at all the clinical trials that occur in the process of developing a new drug up to the point where a drug is getting approval. Now, probably most of the people in this audience are aware that typical drug development involves 3 different phases of drug trials, phase 1, phase 2, and phase 3. But what you might not realize is that when drug developers develop a drug, they typically are testing it not against a single medical indication, but against multiple indications. And so as such, there are often concurrent phase 2, phase 3, phase 1 clinical trial activities, and there are many multiple trajectories within a broader program of drug development, only a fraction of which actually lead to approval. So what I want to do in this empirical exercise, at least, is look at the totality of pre-licensure drug development activities across all indications, across all phases, up to the point where a drug gets approved. My focus is on cancer, and it's primarily not because I'm particularly interested in cancer, but it's because it's the easiest area to work in, because the standards in terms of reporting, in terms of outcome measures, in terms of toxicity reporting, et cetera, are fairly well developed in cancer in contrast with many other disease areas. And to capture all these trials, I've searched many different databases, Embase, PubMed in particular, as well as clinicaltrials.gov. We're only interested in looking at trials up to the point where a drug gets approval, and all subsequent trials that are aimed at extending the indication or extending the FDA label of that drug, and we use strict eligibility criteria, much as you would in any kind of systematic review. And again, my point is that I'm trying to look at moral inefficiencies here, so we need some kind of proxies for moral burdens as well as morally good outputs in drug development. And so what we're looking at when we're extracting elements from these clinical trials for benefit is primarily the number of patients that are exposed to the new drug, also looking at deaths of patients as a burden, drug-related deaths in particular. For benefit, we're looking at enhanced survival in the context of cancer trials, as well as information generated that leads to an FDA approval. And for unusable information, I'm going to describe this later, but basically what this concerns is clinical trials that generate information that is redundant, essentially, with information we already have, or clinical trials that don't really adequately answer the question they set out to resolve. All the extractions we're going to describe to you today involve double extractions. There's a sort of very careful quality control process that we undertake, again, in line with what happens in systematic review and meta-analysis for those of you who are familiar with those areas. Okay, with those sort of methodological preliminaries behind me, now let me describe what it looks like to develop a new cancer drug. And for this, I'm going to focus on a drug called sorafenib, which is a cancer drug that was approved in 2006. And sorafenib is an inhibitor. It was originally developed as an inhibitor of the RAF kinase, but it turned out that its activity is primarily through inhibition of a series of tyrosine kinases. It seems to be primarily implicated in inhibiting angiogenesis. Sorafenib has been approved for 3 different indications: renal cell carcinoma, hepatocellular carcinoma, and irradiation-resistant thyroid cancer. And so we captured all of the clinical trials that were pre-licensure trials of sorafenib, 203 clinical trials, 76% of which were monotherapy, the remainder of which were combination therapy trials. And I'm going to show you in a minute what the translation trajectory looks like using a graphical representation called an arrow diagram. And I first want to kind of give you some orientation with looking at these diagrams. So the phase of clinical trial is indicated by the shape. And if a trial is positive, it's green. If a trial is negative, it's red. And if it's inconclusive, it's yellow. And in the horizontal axis, as you go across the arrow diagram, we're looking at time, so the year in which the trial was launched. And in the vertical axis, we're looking at all the different indications that were tested for this drug, sorafenib. Okay, so this is the start of an arrow diagram, and what you can see here is the very second indication tested, that's hepatocellular carcinoma, eventually led to an approval. The asterisks indicate an FDA approval. And in fact, the third indication tested, renal cell carcinoma, led to the second approval. Actually, strictly speaking, since this stops at the point of approval, RCC was approved before hepatocellular carcinoma. So far, this looks really, really wonderful. Here's another sort of dimension of this arrow diagram, and what you can see is at the point where the phase 3 clinical trial had been launched that led to the approval for RCC, there were only 4 indications that had been tested. This is not an indication up here. And of those 4, 2 of them ultimately received approval. So this looks like a highly efficient drug development process. Now, this is what the entire translation trajectory looked like for sorafenib. What you see is that they picked up an extra indication here under thyroid, but all these other indications were not ultimately successfully reflected on an FDA label. These are all monotherapy studies. This is what happens when you add the combination therapy studies. I'm just going to toggle back and forth so you can see how mono goes first, and then you get a wave of combination trials afterwards. And this is what the entire trajectory looks like if you superimpose the 2 graphs. So the first point I just want you to notice is that there's a huge amount of trialing activity that takes place in the development of a new drug. Now, there are 3 particular dynamics that I want to point out. The first one is that success came extremely early in the case of sorafenib. So the very first 2 out of the first 3 indications tested led to an FDA approval. And in fact, if you look at the total number of patients participating in the whole translation trajectory, that's about 15,000 patients. The fraction of patients that participated in trials up to and including the first indications to get FDA approval is less than half of all the patients that participated. Now, if you look at the fraction of patients in those trajectories that participated in successful translation trajectories, it's actually really quite high. It's somewhere on the order of 80 to 85%. So that looks like an incredible moral efficiency. It looks like for the vast majority of patients that participate in clinical development of sorafenib, up to the point where sorafenib gets licensed, most of the patients are participating in trials that are generating information that can inform healthcare decision-making. And in terms of the number of deaths, this is less than half the number of deaths that occurred in sorafenib development occur up to the point of discovering and vindicating those first two indications that get approval. The second point I want to note is that further exploration of sorafenib activity, either in combination or against various other indications, is mostly fruitless. For all that activity that happens after the approval here, there's only one other indication that's picked up. And so if you look at the fraction of patients that participate in trials that are aimed at extending the label to new indications or combinations for sorafenib, it's about half the fraction of patients are exposed in trials aimed at extending the label. And if you look at the fraction of patients that are participating in successful translation trajectories in those studies that are aimed at extending the label, it's a tiny fraction. So here it looks like we have a high degree of moral inefficiency after licensure. And again, if you look at the fraction of patients that die from drug-related toxicities, well more than half the patients that participate in development of sorafenib die in trials aimed at extending the label rather than get the first label for sorafenib. Now, the good news is, in terms of risk-benefit balance, if you look at survival advantage, it looks like pre-licensure there is a clear survival advantage for patients that participate and are exposed to sorafenib. And post-licensure, it looks like there isn't any survival disadvantage. So despite all that carnage and all that failure, at least we can say it's very comforting to know that participating in trials aimed at extending the license don't necessarily lead to a survival disadvantage for those patients. Now, there's a third point I wanted to note, which is the simultaneity of trials. You see that lots of indications are launched in the same year, and even within certain indications, you have trials that are launched in the same year. So there's a lot of simultaneous research activity. In fact, if you look at the number of trials that are launched each year, you see that the peak of trial launching occurs in 2007 after the first FDA approval of sorafenib. Now, the last point before I move on from sorafenib, it has to do with the amount of unusable information that's generated in developing a new drug. And for that, we came up with a metric called studies of limited value. And what we're measuring there are studies that are duplicative based on 4 pre-specified criteria, studies that lead to positive phase 2 clinical trials, but for which there are no phase 3 trials that are aimed at actually testing and vindicating that treatment using a survival endpoint, and studies that close due to inability to recruit a sufficient sample of patients, defined as 85% of the target recruitment number. And so what you see here is that 24% of the trials, of efficacy trials, phase 2 clinical trials, actually are studies that fall into the category of having limited value. And so if you look at the number of patients that participate in studies of limited value, you see that somewhere on the order of 16% of the patients that participated in trials aimed at developing sorafenib were participating in studies of limited value, and probably about 9% or so of the deaths. So this work was led by my research assistant, James Mantina, who's now gone on to medical school, as well as my postdoc, Spencer Hay, who now has a position at Harvard. Okay, that work is under submission right now. If you're interested in it, you can follow up on a previous article that I published in Journal of National Cancer Institute in 2015 looking at a related drug, sunitinib. That was one drug. What happens when you look at multiple drugs? So for that, we looked at all FDA-approved cancer drugs approved between the years 2005 and 2008. There were 9 different cancer drugs, including sorafenib, and we performed the exact same kind of analysis, and this is what the arrow diagram looks like for all of those drugs. Now, it's a lot of information here, but what I want to point out, again, is that early success happens for pretty much all of the translation trajectories. In fact, if you look at the indication, which indications tested, if you group these together, you see that for about 90% of the time, the first indication tested leads to an FDA approval. For about 60% of the time, 50% of the time, the second indication tested leads to an approval, and then another 10% of the time, the third indication leads to an approval. And you see things kind of flatline after that. You've got this one little blip at 9th indication, but basically all these indications, very few of them, if any of them, lead to an FDA approval. And if you look at the fraction of patients that participate in studies up to and including the first indication getting FDA approval, you see that it's somewhere a little bit more than half of the patients that participate in the whole trajectories, but that a large fraction of these patients are participating in successful translation trajectories up to the point of discovering the first indication. And again, in terms of drug-related deaths, here you have roughly about half of the drug-related deaths occur up to the point where the first indication receives the license. The second point is that further exploration is mostly fruitless. So if you look at the fraction of patients that participated in studies aimed at extending the label across these 9 different drugs, you see that it's a little bit less than half of the fraction of patients in the whole trajectory, but that a tiny fraction of those patients are participating in successful translation trajectories. So again, it's a picture of inefficiency that happens after a drug gets FDA approval, and you see here that about half the the drug-related deaths are occurring in those label extension studies. And the third point is you see a lot of simultaneity across these different drugs. One of these trajectories is sorafenib, but I think this is sunitinib here. And you can see these red strips indicate a lot of trials are being launched at the same time. By the way, these are all monotherapy studies, they're not combination therapy studies. So that work was led by my PhD student, Murph Carlisle, and is right now in manuscript preparation stages. So far, so good. We've looked at what happens and how much patient burden is associated with developing a new cancer drug. What happens when we try to develop a cancer drug and it doesn't ultimately lead to FDA approval? So for that, we created a sample of 9 drugs that got to phase 3 clinical trials, but that ultimately were not approved by the FDA and were abandoned. We used very, very strict criteria to define these drugs. These drugs are from a slightly earlier cohort of drugs than the drugs that I described earlier. We captured 128 clinical trials of these different drugs, and this is what the translation trajectory looks like for all those 9 different drugs. So the first point that I made earlier is that success comes early. Obviously, there's no success with a drug that doesn't get FDA approval, so it's mostly irrelevant, but it is interesting to note that the very first phase 3 clinical trials that are launched usually in the first indication tested. So in only really 2 cases is the first indication tested in phase 3 clinical trials, not the first indication that was actually put into clinical trials in the first place. Interestingly, for only 3 out of the 9 clinical trials, the first phase 3 studies were preceded by positive phase 2 studies. So actually, a large number of these trials were preceded by inconclusive phase 2 studies, or in some cases, there was no phase 2 studies to support them in the first place. Something that surprised me and may surprise many of you in the audience, but that seems to happen quite commonly in drug development. Further exploration is mostly fruitless, well, of course, because these didn't go on to get approval, so that's not at all surprising. But if you look at the number of patients total that participate in development of drugs that get up to phase 3, but that don't get licensed, and you compare that to the fraction of patients that participate in trials aimed at extending the label of already approved cancer drugs, it looks like actually fewer patients participate in these unsuccessful trajectories than in trajectories aimed at extending the label. And likewise, if you look at the number of deaths that occur in trying to develop a new drug that doesn't get an FDA approval, you see that actually fewer patients die in unsuccessful translation trajectories than in translation trajectories that follow after a drug gets approval. Now, unlike in the sorafenib, there actually is a survival disadvantage, as far as we can tell, with participating in trials of cancer drugs that don't get approval. This is just right on the edge of statistical significance. I think it's probably, strictly speaking, I have to call this a trend, not a result, but I think it speaks for itself that there seems to be a survival disadvantage for being allocated to a drug, a cancer drug, that doesn't ultimately get FDA approval, at least for the drugs that we looked at. And then finally, you see some, but not quite as much simultaneity in terms of research efforts. So there's 3 translation trajectories here where you have 5 or more indications being tested at the same time. Now, what fraction of the information generated in unsuccessful translation trajectories is ultimately not usable? We perform the exact same kind of analysis, the limited studies of limited value analysis that I described earlier. And what you see is, in contrast with the number for sorafenib, the number is 13%. So actually, a smaller fraction of patients have limited value for unsuccessfully translated drugs. If you look at how this translates into the fraction of patients participating in the whole trajectory, remember it was around 16% for sorafenib, but for drugs that don't get FDA approval, it's only on the order of about 5% of patients participate in studies of limited value. So it looks like, ironically, there's greater efficiency in developing drugs that ultimately don't go on to get approval than there is in trying to extend the label of drugs that already are FDA approved. So this work was co-led by my PhD student, who I mentioned earlier, as well as my research assistant, Taji Wang. Okay, interesting trends for cancer. Now, again, this is only 3 years of cancer drugs. It'll be interesting to see whether or not these Trends that we've identified hold for a more recent cohort. We're now examining that. But one relevant question is whether or not what we observe in cancer is true of other disease areas. And so we started to look at 7 different areas in neurology as well as pain. And I'm going to present to you some very preliminary data of arrow diagrams from 4 of these different indication, or 3 of these different indication areas. The first is Parkinson's disease. This is a drug called Rationale. And what you see here is there is some further exploration, but the very first indications tested are the ones that lead to FDA approval, and none of the ones that are aimed at extending the label get FDA approval. Same thing for rotigotine. If you look at, that's a Parkinson's disease drugs. If you look at multiple sclerosis drugs for teriflunomide, not very much exploration of other indications, but in fact the only indications really tested are the ones that get approval. And if you look at the drug fingolimod, actually in this case it's the second indication tested that gets the FDA approval. There's some exploration against other kinds of indications like ALS, for example, for the most part not leading to successful FDA approvals. And for the pain drug, we looked at pregabalin. These arrow diagrams are divided into different pain sub-indications or different neurology indications. And what you see here is the first 4 pain indications that were tested ultimately went on to get FDA approval. The first neurological indication tested, partial seizure, goes on to get FDA approval. This further exploratory activity doesn't go on to get FDA approval. And you see in the case of post-surgical pain, a huge, huge amount of activity trying to extend the label for pregabalin to that particular sub-indication of pain. So I think you can say that you see that for neurology and pain drugs, you see similar trends as in cancer with respect to seeing success relatively early on in translation trajectories. You see a fairly robust pattern of unproductive exploration of trying to extend the label of these drugs. And you see some simultaneity, although not quite the same intensivity of simultaneity in trying to develop neurological as well as pain drugs. That work is being pursued by my PhD student, Amanda Hakala, and my other PhD student, Carol Federico. Okay, let me try to summarize what I've shown so far. So the first point is a really good news story. Drug developers are extremely efficient at harnessing preclinical and early clinical trial evidence to get a drug to its first license. Very few patients exposed to get to the point of a license. On the other hand, they're extremely inefficient, it looks like to me at least, in trying to extend the label of an FDA-approved drug to other indications or drug combinations. That actually most of the burden, or at least a large fraction of the burden that occurs in trying to develop a new drug, actually occurs in trying to extend the label of an approved drug rather than in trying to get a drug first approved in the first place. You see a lot of trialing activity occurring simultaneously rather than in a staggered fashion. And you see a large fraction of patients, particularly for FDA-approved drugs, that are participating in trials that actually have very limited clinical value. So what are some of the moral implications of the patterns that I described? Well, I'm just starting to try to work this stuff out. So what I'm going to present now is just some hypotheses that are not fully locked down, but things that I'm exploring. So the first has to do with the way we draw inferences from clinical trials. So this is the translation trajectory for sunitinib, which is a drug closely related to sorafenib. And what you see here, again, is success comes really early. These are the first 2 indications that get approval. Thyroid also gets approval, but you see a sort of desert-like landscape in terms of success with the exception of this one oasis. Now, the response rate in that particular thyroid cancer trial was 31%, which is a very high response rate for a cancer monotherapy clinical trial. And one of the questions that's worth asking is whether or not when you see an oasis amid a desert, whether or not that's reflecting a real drug effect or whether that's reflecting random variation. In fact, subsequent trials of sunitinib against thyroid malignancies showed that the actual response rate is only about 12%. And this raises questions about how we interpret studies that are, when they call themselves statistically significant, when there is so much hypothesis testing that occurs in a translation trajectory. And so my first point is that when we undertake a systematic review or meta-analysis to evaluate pre-licensure drugs, we probably ought to be thinking not merely about the trials testing that exact same indication, but also about trials that are elsewhere in the translation trajectory of that same drug, and that's going to help us to differentiate between random variation and actually a real effect. My second point is that there may very well be a case, this seems like a crazy hypothesis, so it might be wrong, but there may very well be a case for urging actually less testing rather than more testing in terms of extending a drug to new indications in order to protect the interpretability of those trials that show statistical significance in a development program. The second point, set of issues for which I think there are some important implications have to do with human protections. Typically, when we do a clinical trial, we organize it in a way where we stagger risk exposure. So in a phase 1 clinical trial, you might have one dose cohort, you observe the patients before going on to the second dose cohort, et cetera. In a randomized controlled trial, you might have some kind of interim analysis that occurs midway through the trial so that you can decide whether or not to pull the plug before going further on. So what we do in clinical trials is intra-trial cohort staggering. And what I want to suggest is that we might want to think about inter-trial staggering in developing new drugs. So again, you see this highly simultaneous activity across indications and simultaneous activity even within an indication. And it's quite possible we could be achieving the same knowledge about the potential utility of this drug with less patient burden if we were to stagger activities in a more rational way across different indications. Likewise, I think just like we have in randomized controlled trials, stopping rules that indicate when we want to pull the plug on a trial because of untoward safety events, we might also want to have stopping rules for whole portfolios that indicate when we either want to pull the plug in trying to extend the label or at least drive very, very slowly in trying to extend the label of a new drug. The third point about human protections is I actually think there's a very strong case to be made that when we are reviewing trials aimed at extending the label of a new drug to other indications, we probably ought to be demanding particularly strong evidence, given that much of the risk and burden that occurs in developing a drug actually occurs after FDA approval rather than before. And that probably reflects different incentives for drug companies that occur after a drug is approved versus before it's approved. My third point has to do with the resources that we expend in trying to develop new drugs. Recall that I noted that drugs that get to phase 3 but that ultimately aren't licensed actually end up exposing fewer patients than trials that are aimed at extending the label of drugs that already have FDA approval. Now, drug companies often make the case that the reason why drugs are so expensive is because there's so much failure in drug development. And for sure, there is some truth to that. But the fact is, if my findings hold up for other drugs, it looks as if much of the failure, perhaps even most of the failure that occurs in developing a new drug, actually occurs in trying to extend the label of FDA-approved drugs rather than to try to develop a drug in the first place. And I think that really undercuts the argument, for drug companies at least, that some of these high costs of drugs are really justified on the basis of failure in drug development. My last point has to do with the way that we synthesize trial evidence in order to make decisions at the bedside and decisions about what kind of research to pursue. And it has to do with the concept of bandwidth. So when you do a clinical trial, typically the trial is aimed at answering a question about whether or not a drug is clinically useful. But of course, trials are generating all sorts of other information. So you can think of trials as having a very sort of wide bandwidth because they're answering any number of other kinds of questions, including questions that we have about pathophysiology and the pharmacology of a drug. And so it's important that we have systems that are capturing the full bandwidth of information and integrating that into the way we practice healthcare. So let's just go back to the sunitinib translation trajectory. Here you see that there are 2 indications that responded, and you see there's a bunch of other indications that didn't respond. And you might postulate that if there are 2 indications that respond, they probably share certain molecular processes in common with each other, and that those processes are probably not present in the indications that don't respond. And it's through this kind of combining negative and positive information that we can begin to paint a picture about the fundamental pathophysiological processes that are operative in different diseases. Now, that's interesting from a research perspective because, of course, it helps you to plan rationally what trials you pursue. If, in fact, you're getting a bunch of negatives here against these different indications, you might want to be really, really careful before you launch trials against indications that share or that lack the properties of these particular indications. But the point is that a large fraction of what happens at the bedside in cancer care involves off-label prescription, involves prescribing drugs outside the label for which the drug is approved. And much of that prescribing activity is motivated by understanding of basic pathophysiological principles. And so it's really important that we make sure that we are gathering as much information we can in terms of pathophysiology from the constellation of trials that are underway and integrating that into decisions about whether to prescribe a drug off-label. And I would postulate, although I can't yet confirm, this is research that we're undertaking in our lab right now, that one of the patterns that we're going to see when we try to look at the way that negative trial information is integrated into decision-making is we're probably going to see some kind of citation bias where positive trials are cited and they're believed as real, and negative trials are not cited, and probably people attribute all sorts of reasons for getting a negative result other than the fact that the drug doesn't really have activity. Oh, the drug might have worked if we combined it with some other drug, or, oh, we just didn't have a large enough sample size, et cetera. Okay, let me come back to the prologue, which I started this talk. So I made 2 points about limitations with the way that we think about risk in the context of trying to maximize the benefit-risk balance for patients in clinical trials. My first point was that we all have a tendency to think about risk on a per-subject basis and a tendency not as much to think about the number of patients that we expose to risk when we do clinical research. Looking at these translation trajectories, I think, makes it very clear that we probably could be doing a lot better in terms of minimizing total patient exposure to get to the point of an FDA approval. And I've suggested some ways that we might try to operationalize that. For example, instead of simultaneously launching trials against a bunch of different indications, you might want to stagger trial activity, or there's different kinds of staggering approaches that could be used, thereby preventing patients in these indications from necessarily being exposed to a drug. My second point was that when we look at risk-benefit in developing a new drug, typically we're preoccupied with prior trials within that trajectory, and we don't really pay much attention to trials that are in related indications or that are being pursued simultaneously. And occasionally we give a little bit of contemplation to the trials that are likely to follow from that study. So IRBs are really primarily, in looking at phase 2 studies, primarily preoccupied with the prior phase 1 evidence and a little bit thinking about what kind of phase 3 trials might follow. But my point is that IRBs and investigators, when they put their names on clinical trials and begin to recruit patients to them, need to be thinking not merely about the single translation trajectory, the horizontal one in the arrow diagrams, but they also need to be thinking about the concurrent activities across pursuing the exact same drug. So I'm just going to close by thanking my funder, the CIHR, which has funded, at this point now, 2 different grants that are aimed at trying to really understand and improve the risk-benefit dynamics as we develop new drugs. And I'll stop there and take questions. So the question is, the Trump administration is looking to accelerate the process of FDA approval of drugs, and probably I think what they want to do is to lower the evidentiary threshold to getting a drug approved. I think that's probably a reasonable inference based on I don't think I'm putting myself out there by saying that. Okay, so I'm not sure if I really see very much health advantage in approving drugs that are ineffective. So the reason why most drugs fail to get approval is not because the FDA has especially onerous demands. It's because the FDA actually requires evidence of safety and efficacy. So I think the only way to get more drugs approved is to say that we don't really care that much about the efficacy of new drugs or the safety. We're going to allow allow the marketplace to evaluate the safety and efficacy, and I can't see how that could be possibly a good thing. We did have a system before 1962 that approved drugs without requiring efficacy information. I don't think it was a particularly successful system in terms of incentivizing serious drug development and evidence development. So, I guess I can't see a way that vision can be enacted in a way that is supporting and sustaining the incentives to develop truly safe and effective drugs. Yeah, so that's really the sort of the take-home, the sort of the Twitter version of my talk is that much of the research activity undertaken to develop a new drug actually occurs after the drug gets approved, not before. And that's surprising. I think probably most people, I'm guessing in this audience, even clinicians, will probably be surprised to learn that. But that if you actually look at this, and by the way, these translation trajectories are truncated at five years after FDA approval, so there's actually a lot more activity that follows afterwards. So yeah, I mean, I think, from the standpoint of a drug company, once you have a drug approved, you have little to lose by trying to extend the label. And in fact, there may very well be an advantage in trying to undertake these studies aimed at extending the label. I mentioned earlier that in realms like cancer, something like 30 to 50% of prescriptions are off-label. And so one of my hypotheses, and again, I can't confirm this is something I want to test, is that in most areas of medicine, particularly pre-licensure, you have to definitively establish safety and efficacy to get a drug on the market. And that's why we have rigorous phase 3 testing up to the point where you have FDA approval. Once you get a drug approved, if you're talking about a life-threatening illness, you don't actually have to prove that the drug is safe and effective. All you need to do is generate agnosticism within the clinician and patient community such that if they're facing a life-threatening disease, why not give the drug a shot? And so it's quite possible that a lot of this indication extension research, some of it is probably really, I don't doubt that some of it is actually really serious and meritorious research, but to me it's plausible that at least a fraction of that research activity is really aimed at not generating proof of efficacy, but generating agnosticism about the prospect of efficacy.
A while back when I was doing clinical research, the research coordinators at the companies said that we're doing these duplicative studies because it makes it impossible for doctors to adopt the latest drugs without other people encouraging them, and the people who do these trials are the ones who end up encouraging the profession as a whole to adopt this beneficial drug. I'm wondering how much of a role that plays.
Yeah, absolutely. So the question, if try to summarize this, is part of the reason why we might see such intensive research activities is because physicians are reluctant to adopt new treatments. And so if you get physicians to pursue these clinical trials, that's going to help them to assimilate that drug into their clinical practice. And is that sort of an accurate summary of your question, roughly? Okay. So I think it's probably, it's an interesting ethical question. Is it ethical to pursue knowledge-generating activities that prove something beyond what you ought to need to prove merely because doctors don't change their mind quickly enough? That's, I think, sort of what the moral question is. And I would say the answer to that is probably mostly no. I think if, in fact, it is true that physicians are refractory to new information, which I think is not necessarily really that clear. I think the literature is actually a bit ambiguous on that. But if that's true, I think the solution to that problem is helping to get doctors to change their mind in accordance with evidence, not to try to overprove things. So I guess I would be very skeptical of an approach that would sanction this research merely because you're trying to overcome resistance within a clinical community.
Can I ask a question? research can begin to predict whether trials, at least phase 3 trials, some phase 2 trials, are likely to be effective? Is that information that potential human subjects ought to know?
Yeah, I would definitely agree that, well, this is still preliminary. I still would like to see this in a larger number of cancer drugs before I make really strong claims about policy. However, I think it's probably reasonable if patients are being recruited into trials aimed at extending the label, it's probably reasonable for them to know that the prospects of success, of this actually, vindicating its promise, are very slim. I think it's very tempting. When I went into this research, my assumption was that success is going to happen more later in translation trajectories rather than early, because after all, you now know something about the safety of the drug, and you now know something about the pharmacology. So if you ask a naive patient, where are you better off, in the first 5 years of a drug's development or in the second 5 years, I think most patients, including myself before I undertook this research, would say, I'll take the second 5 years. And it may very well be the reverse. And so, yeah, I do think that there is a good case to be made that in informed consent discussions, in the context of studies aimed at extending the label, patients ought to be disabused of any assumption they might have that because the drug already has approval, that it's probably a reasonably safe and likely to demonstrate efficacy.
So along the lines of Dr. Wanger's question, I'm wondering where the locus of moral stewardship ought to be if indeed your research does fully pan out. And so on a simple level, I'm thinking that if an IRB knew that if these extension trials are very unlikely to be effective, then why prove them in the first place? Unless one wants to get at some of the sort of exceptions that perhaps Dr. Corman was getting at or you were getting at a little bit earlier. But it seems to me that's an obvious side of sort of stewardship. But I wonder how you think about that. Do you think that it ought to be a diffused responsibility also?
Well, you're asking exactly the right question, and this is something that I've been preoccupied with ever since I started seeing these dynamics for sorafenib. And I don't have a definitive answer, but I can start to kind of speak around it. So the first point is that I think IRBs have a role to play in a sense that they are the ones that control whether a given trial goes forward or not. But obviously, IRBs have no control over trials that are not within their jurisdiction. I think probably sponsors ought to be expected to, so anytime when you do a clinical trial, you submit it to an IRB or you're showing it to a clinical investigator and trying to recruit them to recruit patients, you'll present them with a protocol or an investigator brochure. And typically that documentation will describe the prior state of evidence leading up to that clinical trial. Now, typically, the information that's contained in that brochure is mostly information about those horizontal trajectories. And what I would suggest is that drug companies and or sponsors ought to be required to present more information about the whole portfolio so that IRBs and others can make better risk-benefit judgments. So I think IRBs have a role to play. I think data safety monitoring boards have another role to play. If you know that there are other trials being undertaken in the exact same indication, you might want to actually have a more sensitive threshold for pulling the plug on a clinical trial because of safety events or futility. Likewise, DSMBs probably should be aware of where the trial is in a whole portfolio, using more sensitive stopping rules later in trials that are aimed at extending the label versus before. I think sponsors probably have a really key role. I doubt that Pfizer or the companies that are pursuing these drugs is unaware of the fact that they have simultaneous drug activities. And I think that there is actually, and I'm not being cynical about drug companies here, I think that they are utility maximizers. They're smart. And I think the rationale for a drug company is, look, before you have approval, you want to concentrate all of your resources in getting the drug approved. So you're going to put your money down completely on those trials that are most likely to get you a toehold in the market. Once you have approval, you can spread your risk. Instead of only testing one indication, you can test 10 or 20 different indications. If one of them, preferably breast cancer or colorectal cancer, relatively common cancers, if one of them is successful, it'll easily underwrite the costs of all those negative ones. So you have a much more actuarial mindset once you have a drug approved. And I think there probably are ways that we could devise policies to try to some extent, discourage a fully actuarial mindset when we try to extend the label. I'm not sure I'm optimistic about these policies being passed in the next 4 years, but nevertheless, I think that there probably are policies we could think about.
I want to thank Dr. Kimmelman for his talk. If you want to hear more from Dr. Kimmelman, he'll be speaking at Cedars at noon tomorrow. And I want to thank Leslie Vermouth, Tom Weinberger for bringing us Dr. Kimmelman today and for their generosity in sponsoring this lectureship. Thank you very much.
Good. I hope it makes sense.
Yeah.
Yeah, right. But you're right. I mean, the point about prediction, I mean,
people are doing the study think it's the best thing left for them. Well, we can talk about
So I one of the things I hope this recording came out because certainly I know one person married. Certainly.
Okay. Excellent.
we're no longer doing they're not no longer doing analysis, just analysis of problems.
Yeah, I think IRBs are, I mean, there's sort of complicated a rock and a hard place. I mean, on the one hand, ethically, you can't evaluate clinical trials without assessing quality. And yet IRBs are harangued again and again for meddling in what some people think is properly presenting science.
So if it's IRB approved, I mean, if it's NIH approved, then what are they getting into? Presumably the science is evaluated. And we have a special committee in the Cancer Institute that does all the scientific review of all the studies, and so the IRB accepts it without any further scientific review. So, but it gets scientific review. So the question is, will those people use the kind of information that you're creating to?
Excellent.
Yeah.
But,
You work on the location too. Yes. Oh, no.
no, I didn't write down.
Yeah, write down the location too. Yeah. Right.
Give me one from Shrimp.
I'm going to go on.
Great.
Very, very interesting.
Stand will be
I
here.
really enjoyed it.
Oh, I'm looking forward to seeing you tonight.
Yeah,
Thanks for
yeah.
your questions. Yeah.
And I know Steve Kimmelman is probably just about to show up. You got the word for the beginning next time.
Yes, that's right. That's right. Yeah. Yeah. Thanks.
And definitely want to.
Oh, good.